Good morning, everyone. Good morning and welcome to this first morning session of today in the Innovation Theater. My name is Agne Alminaite and I work for Thermo Fisher Scientific, and I come from this beautiful Thermo Fisher site in Vilnius, Lithuania. Today I will be talking about isothermal amplification and the reagents we can supply for isothermal amplifications and the new ones we have developed recently. So I will be talking specifically about recombinase polymerase amplification and its uses in research and for diagnostic assays development. So I'll review the technology, the RPA technology. I'll talk about its applications for NGS, for example, and for pathogen amplification, and give a short overview of our other products. So what is isothermal amplification techniques? There are a number of them. You might have heard about LAMP, about helicase-dependent amplification, about rolling cycle amplification. These are the techniques to amplify RNA or DNA without the need of temperature change, therefore without the need for thermocycling. And these techniques are enabled by specific types of polymerases that like to amplify in one specific temperature. These polymerases have one feature in common. They are all strand-displacing polymerases. So while amplifying DNA, they are displacing one strand, and this displaced strand serves as a template for further amplification, making these types of amplifications very efficient and yielding a lot of product in short time. Recombinase polymerase amplification requires several proteins. It's a very similar technique to PCR. It only uses 2 primers and takes place in 37 to 42 degrees and no need to change the temperature. The components or the proteins that are needed for this reaction are recombinase, obviously, and also the strand-displacing polymerase. We used BST DNA polymerase to develop our RPA kit. There's also helper protein and single-strand binding protein. The advantages of RPA as advantages of other isothermal amplification techniques is that they don't require thermocycler. They are fast. RPA reaction gives you results in less than 20 minutes. It's sensitive, specific, robust, meaning it doesn't care about the reaction inhibitors. And there's multiple detection options for the RPA. Therefore, it can be widely used for pathogen amplification and detection, both human, animal, plant, and also in other genetic research, such as incorporating RPA step in NGS, for example. Let's start some data review for pathogen amplification by this method. So on this slide, you see the sensitivity of RPA. So RPA enables amplification from samples containing very low concentrations of target DNA or RNA. On the left, you see the amplification from DNA targets, and Thermo Fisher Scientific RPA kit amplifies DNA from as low as 0.1 picogram of starting material. And on the right, you can see amplification from viral RNA. RPA can amplify from as low as 1 copy of starting RNA. the specificity of RPA. So we are showing here the detection of a single target in a mixture of RNA or DNA mixture. So on the left, you see amplification from RNA mixture of 5 different targets, 5 different viral targets: SARS, Zika, influenza, chikungunya, measles virus, were specifically amplified from a mixture of RNA using the RPA isothermal amplification kit. And on the right, you can see amplification of DNA targets from the mixture. Bacterial and human DNA targets were specifically amplified. As I mentioned, RPA, isothermal amplification, is very tolerant to inhibitors in the reaction, and it gives high performance even in the samples containing these inhibitors. We have tried here several types of inhibitors coming from sample prep reactions like ethanol, from samples itself like blood and urine, and from universal transport media that contains reaction inhibitors. So amplification takes place in the same way as in the reaction without the inhibitors, in those reactions containing the inhibitors. Another very important feature when detecting pathogens is the ability to multiplex. Here we show 3-plex and 4-plex reactions detecting 3 to 4 targets in the same tube from viral RNA or DNA samples. So RPA isothermal amplification gives high specificity and robustness, and you can specifically and successfully amplify multiplex targets without the need for further optimization steps. So just to review the RPA detection workflow, it's starts with RNA or DNA isolation, and we have shown detection from viral, bacterial, human samples, followed by the reaction itself, which takes 20 minutes. It can be a single-plex reaction or a multiplex reaction, and there are multiple ways to detect the result. We have chosen here to show detection on the gel, but fluorescence detection, real-time detection, CRISPR-enhanced detection is also possible. Now I would like to talk about the use of this type of amplification reaction in slightly different application than pathogen identification, and that is about incorporating RPA amplification step into NGS workflows. So NGS workflow starts with nucleic acid preparation followed by library preparation step. An RPA reaction can substitute the library amplification step, which is usually done by PCR, in cycling conditions and higher temperatures, and amplify your libraries in low temperature, which is 37 for RPA, with giving the same results as PCR. So what we have done here is taken 3 bacterial genomes with varying GC content from 33 to 67% of GC, and libraries were prepared from low amount of starting material, and then libraries were amplified using the usual PCR amplification by Colibri amplification master mix or the RPA kit. RPA kit amplified libraries at 37 degrees for 20 minutes. The prepared libraries were then sequenced, and the sequencing results in the table below reveal that RPA and PCR amplification yielded almost identical outcomes. The percentage of aligned reads and the reads with over 30 times coverage are almost identical for when the libraries are amplified by PCR or by RPA. Meaning that RPA is an efficient method for low temperature NGS library amplification. And this may be desired to preserve sequence integrity in lower temperature, and it's also effective amplification of both AC-rich and AT-rich regions, and amplification is relatively short, takes 20 minutes. Thermo Fisher Scientific offers RPA reagents in several formats. So one convenient format is Lie-Ready RPA kit, which is an optimized solution for highly efficient RPA reactions without the further need to optimize. Or we offer standalone RPA proteins, all the proteins RPA needs, plus the reaction buffer, as well as additional components that may be needed for RT RPA, like superscript reverse transcriptase, or RNase inhibitors, DNTPs, primers, and even CRISPR-Cas proteins for sensitive detection. There are a number of other isothermal amplification techniques that we offer products for, such as LAMP, which requires BST DNA polymerase. The RPA I talked about, the RCA, or whole genome amplification technique, for which we have Phi29 polymerase, or the enhanced EquiPhi29 mutant, giving you speed and more yield, and also supplementary products for isothermal amplification. Thank you for coming today. Thermo Fisher is creating value through partnership, and we are always ready to customize our products according to your needs and share our expertise and guarantee security of supply and quality.